In this lecture, we're going to talk about a very important process in the skeleton and the skeletal system. It's called ossification. And ossification simply means to form bone. So our bone actually forms from a material that we're going to talk about today. So in the last lecture, I talked about osteoblasts and osteoclasts. Osteoblasts are the bone-forming cells. They're the immature bone cells, bone-forming cells that produce the matrix that is going to be made up of collagen, elastin, and then also the calcium phosphate in there, and then they eventually become osteocytes, and that's what we're going to talk about today. And there's actually two forms of ossification, so I'm going to go over both of those today, and hopefully it makes some sense to you. So the first type of ossification is called intramembranous ossification. Let me move myself over here so I'm out of the way, hopefully. There we go. All right, so intramembranous means between membranes. So these are going to be what form your flat bones, and I'm going to go into more detail about that. So intramembranous ossification is changing mesenchymal tissue, so this is undifferentiated connective tissue, into bone. So one example would be your skull. Your sternum would be an example, your pelvic bone, your ribs, anything that's a flat bone is going to be formed through intramembranous ossification. So let's talk about that. Basically, you have layers of undifferentiated connective tissue that are going to appear where future bone occurs. So when does this process happen? This process happens when you're an embryo. So when you're first developing, these are the bones that form first in a developing fetus or developing embryo, and this happens during the early stages of development. Your skull actually forms first. That's the first part of your skeletal structure that forms during development. So you get these layers of undifferentiated, so they're not differentiated yet. These are still what we consider stem cells, and they are going to become connective tissue. So they start to appear, they just kind of show up at areas where bone is going to occur. And what ends up happening is these connective tissues, they start to differentiate into these cells called osteoblasts that we just talked about. So those osteoblasts, they're going to basically show up, and where are they going to show up? Where the blood vessels are. That's how they get there, right? So they're going to form around the blood vessels, and they're going to start to build bone. They're going to start to produce their matrix, and they're going to start to encase themselves in this bony material. Eventually, they're going to do what I mentioned before, where they surround themselves in this bone and they become osteocytes. So it's that idea I mentioned before where you paint yourself in the corner, right? So if you paint yourself in the corner, you're going to be in a situation where you can't walk on the wet floor. These osteoblasts produce bone around themselves, and eventually they become encased in that bone, and then they're stuck there and they become osteocytes. But remember, what makes osteocytes different from chondrocytes is osteocytes have those long cellular extensions which produce canaliculi. And what that does is it allows the osteocytes to function as living cells. They can still get their blood because of the fact that they have those canaliculi. And then eventually you're going to get this connective tissue that forms on the outside of the cell called outside of the bone called the periosteum. And the periosteum is that connective tissue layer that is full of blood vessels and nerves. And that is also where the cells are that form new bone. So I like this image I'm about to put up here. This is kind of a cool one. So if you take a look here, what you're seeing is this is a fetus that is 12 weeks into development. And there's two different colors here. The red is bone and the blue is cartilage. So you can see the bone is forming and developing in those spaces. So the blue is still not bone yet, it's cartilage, and then it becomes bone. This next diagram is also a relatively decent one to see what's going on. So in this first slide, what you're seeing is you're seeing osteoblasts, and those osteoblasts are forming in an area where bone will eventually be. All these little cells over here, these are going to be those mesenchymal, these stem cells that appear in those areas. And eventually they start to produce their matrix, right? So these are osteoblasts that are producing their matrix of collagen and elastin. So they're going to, here in the second stage here in this diagram, you can see that they're starting to produce bone, and then the ones that are trapped, the cells that get trapped in that bony matrix, they become osteocytes. And you can see they have those little cell extensions which allow them to get their nutrients that they need. The third stage is at this point we have now formed our spongy bone. So this is that bone that formed here, and you can see blood vessels have started to penetrate the area, which bring more bone cells, and eventually this is what becomes bone. And then in this final stage here, you can see the osteocytes have formed. we can see the periosteum is this outer layer here, and these are osteoblasts, which are laying down bone. And what you end up with is spongy bone in the middle and compact bone to the outside. And the reason why the compact bone forms is because there's nowhere else for those cells to go, so they start to get packed in tight. So when you see a flat bone, the middle of the bone is going to be spongy bone, and then the outer sides are going to actually be compact bone. I'm going to put on a quick little animation video, which I think you'll find helpful.
The development of the skeleton's flat bones begins as certain mesenchymal cells at specific points within embryonic fibrous connective tissue develop into osteoblasts. The osteoblasts cluster together, secrete bone matrix, and develop into osteocytes that deposit calcium and other mineral salts that harden the forming bone matrix. The bony regions spread out into thin columns called trabeculae and form spongy bone tissue. A fibrous covering called the periosteum forms on the surface of the bone, and a layer of compact bone tissue replaces the upper layers of spongy bone. This process in which flat bones develop from connective tissue is called intramembranous ossification.
So that video was a quick overview of what happens during intramembranous. So what do you need to know about intramembranous? This is how flat bones form. So flat bones form through this process, and you get a scenario where osteoblasts form bone in the center, they spread outwards, and it eventually becomes bone. The second type is a little more involved, and the second type is called endochondral ossification. And endochondral is how the rest of the skeleton forms. And this image shows you a developing fetus. This is actually an x-ray of a developing fetus, and you can see these little bones here. Those are eventually going to become bones. Now, endochondral is a lot more involved. This is how our long bones form, our short bones, our irregular bones. So I'm going to go through this, and then we'll see how we're doing. Okay, so here's a big difference. In intramembranous bone, it's not cartilage yet. It starts off as this mesenchymal stem cell that becomes flat bone. This is a big difference. Endochondral bones start off as cartilage. So this is a fully formed cartilage mini skeleton. So it's a cartilage model that forms at the future sites of bones. So in this image here, you can see this is the radius and ulna, but they're not a full bone yet. Those are actually made of cartilage, and they're going to become bone. So one big difference between intramembranous and endochondral is endochondral starts off as a mini version of the bone that is going to be cartilage. What ends up happening is cartilage does what it does, and it builds lacunae, and it dies. So it leaves behind these open rooms like lacunae, called lacunae, I should say. What ends up happening is the osteoblasts move into those empty rooms. So this is kind of dark, but let's say somebody died and their house was left empty. Eventually, someone new is going to move into that house, right? So they're going to move in and make that house their own. That is essentially what osteoblasts do. The cartilage cells live for a little while. They make these little bony models, and then they die. And when they die, they leave these open room called lacunae, and then the osteoblasts are going to move into that space, and they're going to start to form bone. The osteoblasts eventually, just like before, they form this matrix around themselves, and they become osteocytes. So just like in intramembranous, osteoblasts become osteocytes when they become embedded in the matrix. The big difference here is osteoblasts start forming on cartilage in intramembranous. And eventually they become surrounded by a bony matrix. The second big difference, this shows you a little diagram here of how that works. So you start with your hyaline model, those cartilage cells die off, and what ends up happening is ossification starts to occur, and you get the bony material forming first in the middle and then in the epiphysis. So it starts in the diaphysis and it ends in the epiphysis. And then there's a secondary ossification center, which I'm going to explain in a moment. And then finally, you end up with your full bone with only one spot having cartilage still. So I'm going to explain that better. So let's talk about that now. Here's another big difference between intramembranous and endochondral. Intramembranous ossification has two stages to it. Intramembranous ossification has one stage to it. Endochondral ossification has two stages to it. It has a primary ossification center, and it has a secondary ossification center. And this is very important, and this is what allows us to do something that is very important to us, and that is grow. So let me explain it. In a long bone, that cartilage gets replaced by bony tissue. It starts in the diaphysis, so it's the diaphysis first. That is called the primary ossification center. After a little while of development, then you get a secondary ossification that happens in the epiphysis. And what you end up with is a scenario where you have the diaphysis first and then the epiphysis. And you end up with a scenario where you get a thin band of cartilage that gets left behind. So I want to explain that a little bit better with a diagram. Okay, so let me just go ahead and do a little drawing here. So let's say I have my cartilage bone here. So this is not bone yet. This is a cartilage model, and we're doing endochondral ossification. What ends up happening is there's a primary ossification center. Blood vessels penetrate this cartilage, and they bring osteoblasts. And what the osteoblasts do is they start to convert this into bone. So what ends up happening is in the primary ossification center, the primary ossification center happens first in the diaphysis, and you get the diaphysis forming bone. And then right before birth, this actually happens right at the end of your development, there's a secondary ossification center that happens in the epiphyses, and you end up getting more bone forming at the ends. But why is this such a big deal? Well, what you end up with is this small band of cartilage that stays behind. So we get a little band of cartilage right where the diaphysis and the epiphysis meet. And what is this small band of cartilage, you may ask? That is going to be the epiphyseal plate, or your growth plate. Why is this so stinking important? Because by leaving this little bit of cartilage in the middle here, that allows us to grow. And there's also going to be a little cartilage left at the end, which is going to be what we call our articulating cartilage. So secondary ossification is very specialized for one thing, for developing long bones that can still grow. If it all just happened at once, let's say there was just one ossification center, the whole entire bone would form and you would never grow. So by having two ossification centers, that allows your bone to develop and grow over your lifetime. Eventually, when you guys hit about 25 years old, that's about the age where this is going to eventually become bone. So eventually, your ossification centers are going to fuse, and this whole thing is going to be bone, and you can't grow anymore. And the only cartilage you're going to have left is going to be at the ends, and that's the articulating cartilage. So I'm going to show you one more short video clip that I think will hopefully explain this. And that's going to be the endochondral. It's good music, right? I'm into it.
During embryonic development, cells called chondroblasts begin secreting cartilage that develops in the shape of the skeleton's long bones. The bluish, transparent avascular tissue formed is called hyaline cartilage. As the cartilage grows, the chondroblasts in the interior calcify and die. Blood vessels penetrate the structure and deliver osteoblasts. These cells lay down bone material. As the cartilage ossifies and spongy bone forms, a medullary cavity develops. At the ends of a long bone, ossification from cartilage also occurs. This process, in which hyaline cartilage becomes bone and grows, is called endochondral ossification. Bone growth continues through childhood. After puberty, bone tissue replaces the remaining hyaline cartilage, and the shaft reaches its adult length.
So just to review, endochondral ossification starts as a hyaline cartilage model. You get your primary ossification center first, which forms the diaphysis. You have your secondary ossification in the epiphyses, and what you get left with is this thin band called the epiphyseal plate, which allows for growth, and the articulating cartilage at the end. Eventually, when you hit about 20, 25, that actually becomes bone as well, and you no longer grow. So what are the differences? Intramembranous forms flat bones, and it happens in one step. There's no secondary ossification. So your flat bone forms spongy bone in the middle, compact to the outside, and that is going to be intramembranous, and it starts from mesenchymal cells. Endochondral, on the other hand, starts from hyaline cartilage, and it becomes bone in two steps: primary ossification and secondary ossification. And that is so important because it allows our long bones to grow. If it just happened in one step, our long bones wouldn't grow. Thank you very much.